Hello, everyone. I'm Tony Hu. I'm just waiting for someone to introduce me. Yeah, yeah. I'm the presidential chair in the biotechnology innovations at Tulane School of Medicine, and it's just moving so fast. So sorry, I have to wear the mask again. And a few weeks ago, I got the flu, and and also three children at home. So I got everybody in trouble. So our lab has been working on the tuberculosis and some cancer disease too. Why we want to focus on TB is because before COVID, TB is definitely the number one infection disease in the world. And it's still infecting the 1/3 populations. And the last week, WHO just issued the TB report 2022, and the death rate increases 3.7%. That's significant. And also the drug resistant also increased. Why? Because there's a lot of the case got delayed due to the pandemic, and also after this okay, all right. So the TB diagnosis for pediatric is still very challenging, and the current gold standard still remain around about 20%. But TB is really different from the COVID, right? The COVID, we just know it, but TB, we know this pathogen for centuries. But some expert state the TB is most successful pathogen in the world. And but why is it so challenging to diagnose? Because we never change the concept. We always rely on the use of a sputum, and we take the sputum from the patients and they culture them. That's the current gold standard. This method it takes a very long turnaround time, generally 6 to 8 weeks. And also, no matter how sensitive they are, they can only cover 50% of patients. Why? Because there's a lot of patients, they're bacterial, not stay in the pulmonary. They are extrapulmonary TB. And also for the HIV infected patients, and you cannot produce the sputum very well, and also pediatrics. So the NIH also promote the new technology, which is called the Gene Expert. This counting the DNA profile still in the sputum. So even it's faster, but it didn't change the landscape. So back to the 2015 on the conference in Washington, DC, I attend. The Tony Fauci also gave the highest priority for TB diagnosis, which is rapid and independent of a culture, and do not use the sputum anymore, and also which can provide quantitative information in tracking the treatment efficacy, because TB drug resistant is also the problem. And our lab for the last 10 years, and we have been focusing on the multiple markers, multiple species in the blood. Okay, so think about it. If you can capture the biomarker to diagnose active TB from blood, you don't need to care where the bacteria are located, pulmonary or extrapulmonary, and you don't need to care the age, senior or pediatric, and also their HIV status. So we're starting from the antigen peptide and then work on the extracellular vesicles. And also recently we have the new paper published on the Lancet that focusing on the cell-free DNA detection from the blood. All these spaces actually representing the different biological pathological situation for the patients. As an engineer team, we're just trying to provide the best tool for the scientists and also the industry partners and also the doctors in the clinic. One of our company actually also located in the Novick is called the Nanotechnologies, and already developed the antigen peptide platform rely on the use of a mass spec, and it really can reach the antigen, the MTB antigen in the blood at 0.01 picomolar. This is really the lowest concentration mass spec can reach. And so this technology, starting from the 2013, we just first time to have this idea. Then we work with a lot of collaborators worldwide. And then the last year, we just got the FDA's recognition as a breakthrough technology, and also got SBIR phase 2 funding last year. And so right now, the company is ready to start the clinical trial in Miami and also Houston. Uh, yeah, so but the second species we like to emphasize is extracellular vesicles. That's yeah, this new generation of biomarker has been applied to the many disease diagnosis, and so they're generating in the cell, in the infected cell, and they carry a lot of original informations, and then they have a chance to flow into the circulations. And once if you have the method to try to capture them specifically and recognize which this exosome, this extracellular vesicle come from the what cells, and you will be able to provide the direct evidence for disease diagnosis. But how to specifically capture them? So we rely on the use of the surface antigens, and so when we checked the TB, and we were still targeting on TB, when we checked the previous literatures, and we found out that there is a very interesting the studies conducted by the group at Stanford. So they identified if you can quantify the LAM and LPRG, these two markers, and they understand their interactive behavior. So you can really differentiate the TB at the active status and from the latency. So that's perfect because then our hypothesis is if we can co-profile these two markers on the vesicle membrane, and we should be able to generate the straightforward method for the TB diagnosis. So how to profile this two? Because we also need another markers to capture the vesicle. And there's another two markers to capture LPRG and the LAM, okay? And then, sometimes the idea is just generated in anywhere around your life. And Home Depot has the children program. And you know that, right? So every weekend, and you brought children there, and they got a free piece, and they can paint, they can build up the house, and they painted the color on it. And when my son at 8 years old, and they mix the paint in the green color and the red color, they find out it's come to yellow. And he asked me what's going on here, and I use my very limited knowledge in the physics to explain that's caused by the overlap of the wavelengths from the paint. It seems like he doesn't understand. But after coming to the lab, I think it could be served for capturing MTB specific extracellular vesicle. And because we know, I have the background in the nanoengineer, so we know the nanoparticle at the sphere shape, they're showing the green color, and the nanorod showing the red color. So when they get close enough, like within the 200 nanometer, that's exactly the single vesicle size, and they can showing the color change. Okay, so I asked the postdoc to do the proof of concept experiment, and they successfully show it works. So then we can we apply them to the TB. Then we also think about so here is another strength from our team. We always think about what's the need for the clinical lab. And if we just spike the sample one by one and running them, and the clinical lab, they won't use it. Then we designed the software to convert the near field microscope to the far field microscope and enable the high throughput scanning. And so that will require some algorithm, the programming, and so we introduced the machine learning the technologies, and try to reduce the noise and capture the real signal from the MTB derived vesicles. And then right now the technology has been the high throughput, automated, and easy to handle. And then we also validate our technology using the confirmed TB and confirmed control. And the sensitivity we can reach is 95%, specificity is 94%. Then we challenge this technology to the most, we call it a triple threat in the field, which is a HIV, TB co-infected children. Okay, so yeah, so this is really probably the most challenging cohort in the world for TB diagnosis. And so far, most technology can only reach like 5%. So we compared our technology with the gold standard. And so there's three groups, like the confirmed, unconfirmed. So confirm that means that the cases can be identified using the current gold standard. And so just look at this, and okay, we're better. Okay, we're better here. Okay. Yeah. Okay. So again, thank you so much for NIH sponsors, and you can tell we generate a platform technology. It's not only for TB, for cancer, for many other diseases, and so just simply change the antibody, and you can really serve many other technology the need in the clinic. Thank you so much. Yeah.